So you're ready, Natalie? Okay.
Yep.
So hello and welcome, everyone. Thank you for joining us today. We are in the process of wrapping up the Summer MS lecture series. This was very successful, very well received, very well attended. And today and over the next week, we will have summer student presentations. And these summer students are supported by the Alberta MS Network. So today we will start with Natalie Tilbury. Natalie has just completed her third year in BS in Neuroscience program at the University of Calgary. This is her second year working with Dr. Alexander McGirr, and she's working, she's investigating the effects of Ibudilast on disease progression in EA model of MS. So thank you, Natalie, and over to you.
Awesome. Thank you for the introduction. That was great. Yeah, so again, my project is the effects of Ibudilast on disease progression, behavior, and microglia in an EAE model of MS. And I added microglia in italics here just because that wasn't in my title before, but it should have been because that was kind of my main focus for this summer. So I'll just go over briefly what I'll be talking about today. So I'm going to give a brief background of the project and kind of why we did what we did. And then I'll share the pilot data that we collected last summer on the disease progression and the behavior portion of the project. And then a little bit of data I was able to gather this summer regarding microglia. And finally, I'll end with some future directions. So to start off, obviously this audience doesn't need much of an introduction to MS. But I did want to emphasize the vast heterogeneity involved in the disease. So even if you look at the main hallmarks, inflammation, demyelination, neurodegeneration, and gliosis, already there's a lot going on there and a lot of interactions between the immune system and the central nervous system. We also know that MS can present in multiple different disease courses, which are distinct in their presentation, and so that adds to that heterogeneity. But what's important to note is that in addition to the sensory and motor impairments that are characteristic of MS, about 40 to 60% of patients also experience neuropsychiatric symptoms such as anxiety, depression, memory loss, and other cognitive impairments. So for example, this graph on the right here shows the percent of patients in a large database with different neurological diseases that develop depression. So as you can see, MS is pretty high there. And these symptoms, therefore, are obviously pretty important to address in MS pathology. So we also see these neuropsychiatric symptoms in EAE, which is a pretty effective model of the inflammation and the immune cell activation seen in MS. With this model, we get a progressive ascending paralysis beginning around day 9 post-immunization, and usually reaching a peak around day 17, but that can be variable sometimes. In terms of the neuropsychiatric symptoms that we see, you get anxiety and depression-like behaviors, as well as cognitive deficits in this model. And it's been previously shown that these symptoms begin around day 6 post-immunization, so they precede the onset of motor impairments. Figures on the right here are taken from a study that tested these behaviors in EAE versus control mice in between days 6 and 8 post-immunization, so before that motor symptom onset. And at the top you have tests of anxiety-like behavior, so the elevated plus maze and the open field test. And then on the bottom you have tests of depression-like behavior, so forced swim, tail suspension, and social interaction tests. And all of these tests show an increase in these anxiety and depression-like behaviors in the EAE animals compared to the controls. So for example, the EAE mice spent much more time in the closed arms in the elevated plus maze, which demonstrates anxiety-like behavior. And the EAE mice were also quicker to become immobile in the forced swim test, which indicates depressive behavior. So we have these neuropsychiatric symptoms presenting pretty prominently in EAE. What we want to do is find a way to address these symptoms and possibly improve them while potentially benefiting the other impairments involved in EAE at the same time. So it turns out there's a family of molecules that looks pretty promising for that, and that's phosphodiesterase 4 or PDE4 inhibitors. So PDE4s, first of all, are enzymes that are highly expressed in immune cells and in the central nervous system, particularly in parts of it that are vulnerable to the effects of MS. PDE4s hydrolyze cyclic AMP, which is involved in many different signaling pathways. And cyclic AMP hydrolysis has a range of effects, but a few of them are highlighted here. So most notably, immune cell activation and migration, inflammatory responses, and neuropsychiatric symptoms. So when PDE4 is inhibited, for example, by the PDE4 inhibitor Ibudilast, these pathways are inhibited and the resulting effects should be decreased, leading to reduced immune response and inflammation, and importantly, a reduction in those neuropsychiatric symptoms. So PDE4 inhibitors have been researched for the treatment of depression and have also been shown to have pro-cognitive and anxiolytic effects. So this makes them an excellent candidate for targeting those neuropsychiatric symptoms in EAE. So as I mentioned before, Ibudilast is one of those PDE4 inhibitors, and it actually has already shown some beneficial effects in EAE and in some forms of MS. So first, one study showed Ibudilast was able to ameliorate disease progression and reduce spinal cord immune cell infiltration in EAE in rats compared to a placebo. So that's on the left here. And the bottom graph, which is from that paper, shows the disease progression using the clinical score over the course of the disease. And as you can see, they used a low dose and a high dose of Ibudilast. And you can see that the high dose of 10 milligrams per kilogram was effective in reducing the severity of the disease course. And then the images up here, they illustrate the reduced cell infiltration into the spinal cord with Ibudilast, and that's using Luxel Fast Blue and H&E staining. And then on the right here, this is a graph from the phase 2 clinical trial of Ibudilast in progressive MS, where it was able to reduce whole brain atrophy compared to placebo over the course of the study. And the graph shows a smaller change in brain parenchymal fraction, which is a measure of brain atrophy. So not only is Ibudilast a PDE4 inhibitor, which are known to have those beneficial effects on neuropsychiatric symptoms, but it's already shown to have some benefits in EAE and in MS. So again, since we know that PDE4s are highly expressed in both the immune system and the central nervous system, and that PDE4 inhibitors have been shown to have pro-cognitive and anxiolytic effects, and as well, since Ibudilast specifically has been found to have benefits in EAE and MS, we therefore hypothesize that Ibudilast may ameliorate the neuropsychiatric symptoms seen in EAE, while potentially having beneficial effects on the disease progression as well. So in order to test this, we induced EAE in female mice using myelin oligodendrocyte glycoprotein and CFA, and the controls received only CFA. We administered 10 milligrams per kilogram of Ibudilast daily to one of the EAE groups, starting from the day of immunization, and the other EAE group and the CFA group received vehicle or placebo. And then we completed behavioral testing between days 6 and 9 prior to motor symptom onset, and we scored the disease progression daily on a scale of 1 to 5. And then we collected tissue samples on day 17 and prepared them for imaging using immunohistochemistry. So the first behavioral test that we did was the elevated plus maze, and this is again a test of anxiety-like behavior. And we found that Ibudilast treated animals spent more time in the open arms of the elevated plus maze than the EAE animals. And so that's suggesting an improvement in the anxiety-like behavior with Ibudilast, which is a pretty promising result. Later on in our behavioral testing, we did the novel object recognition test, and this is a test of hippocampal memory. And so in this, mice are introduced to a few identical objects and then tested for their preference for one of the familiar objects versus a novel object. So that's shown in the diagram on the left here. And we found in this test, there was no difference in the time spent investigating the novel object for each group. However, there was quite a bit more variance in the Ibudilast treated group compared to the EAE and control groups, as you can see on this graph here. And so this is what I mean by disorganization in hippocampal memory behavior. The scores are all over the place. There's not really a clear trend. And we think this could be explained by the possibility that these animals were beginning to get sick at this stage, which is plausible when we take a look at the next slide. So this was our most striking finding, and it was that Ibudilast actually accelerated the motor disease progression of EAE. So on the left, you see the change in weight over the course of the disease with the Ibudilast treated animals in red, and you can see that there's a pretty large drop in weight for those animals compared to the other groups. And then on the right is the clinical score, and again, you can see that the red, the Ibudilast treated group had a large increase in score earlier in the disease course. So it's not shown here, but we know from a couple of the other cohorts that we did that the EAE animals that were untreated were getting sicker around day 20. So these Ibudilast animals were having a large increase earlier in the disease course. So this is obviously very unexpected when considering our hypothesis. So we wanted to investigate these findings a little further, and the next step in order to do this was to characterize these findings at the cellular level. And to do that, we started with microglia. We wanted to look at microglia because they are the resident macrophages of the brain, so they're kind of a marker of inflammation, and they play a large role in the inflammation and demyelination in MS. So when microglia are activated, they can promote inflammation by releasing cytokines and cytotoxic factors and interacting with other immune cells. And so this is associated with the demyelination. So therefore, microglial activation can be detrimental in an inflammatory state like in MS. So from our pilot data, we further hypothesized that Ibudilast treatment may be associated with an increase in demyelination, as demonstrated by that accelerated disease progression, and that this increase in demyelination may be related to increases in the number and activation of microglia in the CNS. So I used immunohistochemistry to look at microglia in the hippocampus in the CA1 region, and as well, I also looked at the gray and white matter of the spinal cord. And so these are representative images in the hippocampus demonstrating microglia in the EAE on the left, the EAE group versus the Ibudilast treated animals on the right, and then some general differences you can see in their morphology here. So we did find an increase in the number of microglia in the region of interest that we used for the Ibudilast treated animals compared to the untreated EAE animals. So that was in the hippocampus and shown in this graph here. So you can see there's an increase in the microglia count. And then for the spinal cord gray matter, again, these are some representative images. And you can see on the left EAE again versus the Ibudilast treated animals and the differences in morphology here as well. And we also found an increase in microglia count in the gray matter in the Ibudilast treated animals as well. So similar to the hippocampus findings, there was an increase in the microglia count in the spinal cord gray matter. And I'm currently working on the data for the white matter as well as intensity quantifications, but I've had some issues with the microscope and with COVID, so unfortunately couldn't present that data today. But to conclude with what we did see, firstly, we saw that Ibudilast may help ameliorate neuropsychiatric symptoms such as anxiety in EAE, but that Ibudilast treated animals' behavior was largely disorganized in other tests. We saw that, strikingly, EAE disease progression was accelerated by Ibudilast, and this was accompanied by an increase in microglia count in the hippocampus and spinal cord in those Ibudilast treated animals. So in terms of some future directions, first and foremost, I'll be working to complete the microglia quantifications I mentioned, and I'll also be characterizing demyelination in these groups using Luxol fast blue staining and cell infiltration using H&E staining. Along with this, comparing these findings to characteristics of microglia in non-EAE mice as a control, as well as non-EAE mice treated with Ibudilast, could be beneficial to expand our understanding of Ibudilast effects. And doing these characterizations in groups prior to motor symptom onset would be beneficial in understanding the cellular basis of the neuropsychiatric symptoms and how they're affected by Ibudilast. So yeah, that's all I had. And I'll look at the chat box for any questions now. So I'm just looking at the chat box here. Did you notice neuronal cell death in hippocampus and spinal cord that might have resulted in the more microglia activation? So that'll be what I'm looking at with, sorry, not neuronal cell death, but I'll be looking at demyelination in those areas as well. So that'll kind of give us a more complete picture of what's going on. I think it would be interesting to look at other cell types as well, immune cells and other cell types in these areas, just to see what kind of different interactions are going on, because we know a lot of those cells interact with each other in EAE.
Do we have more questions from audience? You can just unmute yourself and ask a question. Yeah, no. Okay. Yeah, so we'll move forward with our second presentation. So Natalie, if you can stop sharing. So Kaylyn, will you start sharing your presentation? Kaylyn, are you there?
Hello?
Yeah, hi Kaylyn. Will you start sharing your presentation now?
Yes, absolutely.
We do have one more question for Natalie if she
Oh yeah.
will.
Oh yeah.
Answer.
Sure, yeah.
So the marker that we used for microglia, we used IBA1.
Thank you, Natalie.
So Natalie, thank you so much. It was a great presentation. And Kaylyn,
Hi.
are you working fine?
I'm just having a little bit of issue trying to get your screen. Or trying to pull it back up. My apologies.
Okay, meanwhile you work on it, I will just introduce you. So Kaylyn has just completed her 4th year in microbiology at the University of Calgary. This is her 4th year studying MS in the context of immunity with Dr. Shalina Usman. And she is going to talk about her work on mass cytometry to elucidate novel lymphoid phenotypes in EAE model.
Okay, I can't quite see you guys. I really hope that you guys can all see me.
Yes, we are seeing you well. We can see your presentation. Just put it on presentation mode. Yeah.
Perfect.
Perfect. Yeah.
Okay, so hello everyone. Thank you so much for coming to listen to my project. I've been again with Dr. Shalina Usman for the last few years working on mass cytometry to elucidate novel lymphoid phenotypes in experimental autoimmune encephalomyelitis. So just a little bit of background on immunology and autoimmunity. Originally, it was thought that the CNS was immune privileged. However, it is now known that some immune cells can infiltrate, which causes inflammation. Albeit there is a limited knowledge regarding the different types of immune infiltrates. However, it should be noted that, again, naive CNS tissue will have fewer immune cells, and that throughout the disease course of many neurological diseases, such as multiple sclerosis, that immune cell quantity and phenotypes can change. What I decided to focus on for my presentation are mostly lymphoid types of cells, such as T cells, some B cells, and things like that. So a little bit of background on multiple sclerosis. Multiple sclerosis, or MS, is believed to be an autoimmune disease that affects the central nervous system. There are three main types: primary progressive, secondary progressive, and relapsing remitting. And relapsing remitting is what I decided to work with. It is characterized by a few important traits, such as inflammation that leads to CNS tissue damage, demyelination, which can result in the destruction of the myelin sheath or oligodendrocyte body, degeneration and scarification of axons, and gliosis. Currently, the overall cause of MS is unknown. So, one thing that I really liked, I went to the MS Network presentation last January, and Kevin Thornburg used this to explain MS and immunity. So sometimes we will have our little myelin with our axon, which is our hot dog in our bun. And we'll get some kind of crazy immune cells come in, and they try and, degenerate and get rid of our myelin. Luckily, we do have some sort of cells, such as astrocytes, that can come in and try and yell at that dog, which is our immune cells, and get him to leave to try and help save our myelin. And this is a concept that I found very interesting to me, because there's a lot of research on why things start to get bad in relapsing remitting MS, what happens when it's bad, but no one really looks at why things start to get better, which is what I wanted to do for my research. So I decided to employ a mouse model known as experimental autoimmune encephalomyelitis. And I wanted to look at naive spinal cord, pre-symptomatic, peak, and post-symptomatic phases. And I just believed that there will be an inflammatory infiltrate that will enter into the spinal cord before clinical symptoms arise of EAE, that later on in disease, autoreactive lymphocytes will infiltrate along with other effector cells. And in remission, I think that phenotypically novel immune cells with possible anti-inflammatory properties will appear, which can lead to the dissipation of the inflammatory infiltrate. So for my objectives, I mainly wanted to examine the roles of the different lymphocyte populations and what their overall role is in the spinal cord during EAE. And so my first aim is to elucidate the various immune phenotypes present in EAE using CyTOF. So mass cytometry, otherwise known as CyTOF, is a newer method to look at cells. And in the past, we have used methods such as flow cytometry to identify cells of interest. However, this isn't necessarily good for exploring the different types of cells that are present, because you are limited by things such as bias, and then you have to compensate. So if you look at the bottom left, I don't know if you can see my cursor, there is a flow cytometry data. And if you look, as you add more and more panels and different colors, the wavelengths start to intersect, which can cause muddled response, and you're not really sure where these are from, which leads to compensation, or a need for compensation. CyTOF, however, allows for the detection of up to 100 different parameters using heavy metal isotopes, and is well suited for my research in an exploratory manner. And basically what I'm doing with CyTOF is I'm trying to play guess who. Instead of having to say, oh, I think this will be here and this will be here, and I only have a few guesses, which will not allow me to find those specific phenotypes, I can actually I'm using a panel of about 32 antibodies, I believe, in order to kind of figure out what types of cells are present, and that way I can further differentiate. A little bit about the workings of the CyTOF. First, you will take your cells, you harvest them, you label the antibodies. They will then be sent through a nebulizer. These are then atomized and ionized, but so is everything else, such as the argon from plasma, carbon, nitrogen, and oxygen. So they are sent through quadrupoles to help filter out the overly abundant ions, which are light ions. That way only the heavy ions go through and is run through a time of flight, separated, and then we are able to analyze our data. Sometimes what happens is you end up with rain plots before you can analyze the data. And this one is just a very simple one that I have. I have a couple stains. I'll draw your attention to the very first line, which is a CD45. And this is just a rain plot that will filter down like this for all of the events. I usually use about 300,000 events per sample, and this way I can just kind of figure out what's there. On the other side, I do have some beads as well, just to help. One thing that can happen though is you can end up with, they're called tire tracks. And if you look in the middle of the screen, there are a few big black blobs. And that happens when there's some sort of contamination, which is why we like to use the rain plot, because then you can go through, filter all of that out, and that way it won't be included in your results. So for me, I would induce EAE. On days 0, I would inject a myelin oligodendrocyte glycoprotein mixed with complete Freud's adjuvant. And I would do that in, I would do an intraperitoneal injection, sorry, a subcutaneous injection. And then for my pertussis, I would inject on day 0 and day 2, doing a tailbane injection. I would then score on a 5-point scale, which I have included in my next figure, which will say score and symptoms, 0 being asymptomatic, 1 limb tail, 2 partial hind, 3 full hind limb paralysis, 4 partial front limb paralysis, and 5 is usually around death. We try not to keep our mice around there. And I have an example set of what the scores will look like. And then on day 6, 12, and 25, I'll harvest the spinal cord for the mass cytometer. My second aim, after taking all of my samples, is I then want to look for any populations that may have healing or anti-inflammatory properties using advanced data analyses. So after the run, I would then take my cells and gate them through cytobank. With flow cytometry, you do a lot of manual gating, but as there are so many more markers, if you continue to manually gate, with mass cytometry, then it will introduce a lot of bias. Therefore, we just try and gate down so that we have live singlet immune cells. So we just gate for their CD45 positive, which will just label all of our immune cells. And then I use a dimensionality reduction analysis known as VISNI. This will then be followed by a self-organizing map algorithm known as FlowSOM. And then data was analyzed using equal number of events with an N equals 14,128, which are randomly selected. So again, our VISNI analysis is a dimensionality reduction analysis, and it allows for permutation of all the markers in cells. You can analyze them equally or proportionally. I chose to analyze them equally, just as, again, I've mentioned, it's very hard to get some of those higher numbers of immune cells in a naive spinal cord or even a pre-symptomatic. So I didn't want to miss any cells by analyzing it proportionally. And this will allow for the most unbiased clusters you can have. Unfortunately, there was a contamination in my pre-symptomatic, so I was unable to replicate the data as well. And this is an example of what my VISNI analysis will look like. From here, this looks very cool, but what do you do with it? You have to try and then pick out different groups here. So what I've done is I've gone through and I've employed just a colored clustering method on it. And then from here, I would then run my FlowSOM data. And this is just clustering all of that data that you saw in the previous slide into observable groups known as metaclusters. And these are my samples currently. In the top, we have our peak. In the bottom, we have our post. And from the data, we found that there are about 20 different metaclusters. For example, you can get different types of images. If you look at post F, where the red circle is, sometimes you can get little clusters that are either really small or they look separated, and you're not too sure about them at first, so you have to go through and just kind of try and analyze your data. What I've done is I was trying to learn, Dr. Joel Sisman, I believe, is trying to teach me how to concatenate my data, which I haven't quite mastered, so I've tried to include on the right what it would look like. And this is just kind of clustering everything into these observable groups. And I have highlighted each cluster from the right and circled them with the corresponding cluster or the corresponding color on the left. The next thing I've done, because that looks wonderful, but to just try and put it in a more like visually like understandable method, I've used heat maps. So these are all of my samples in my heat maps that I have tried to look at. But for the purpose of this presentation, I'm just going to look at a single one. Oh, sorry. Anyways, after analyzing, I got these two slides mixed up, my apologies. After analyzing, I went through and looked at all of the markers and how high or low they were and tried to group them into either peak disease or a post disease. And these mean that if it's in peak disease, we had higher numbers or levels in the peak samples. And if it's in post disease, it just means that we had higher numbers present in our remission phases. So all of the ones that I have in red boxes are what were higher in the remission phase, just as that's what I wanted to look at. All of these samples that now have the red arrows next to them are what I believe to be some sort of T cells. And CD3E is what is supposed to be used to stain, to indicate different types of T cells. However, one thing I found interesting is when you break it down again, CD4 is our helper T cells and CD8 is our cytotoxic T cells. And if you look at metacluster 15, we see that it's staining for both CD4, CD45R, which is a B2, which is also B220, which is a classical B cell marker, but there's no staining showing up for this CD3E. I went through and decided to try and check to see if this occurred in every single sample. And it was still showing up as no CD3E, but CD4 and CD45 are presenting. So I knew it wasn't just an error in the example, which I have highlighted here. I tried to up the scale as well, just to see if it would break down even further. And I've noticed that the remission phase has a lower level of the CD4, but it's still both presenting. So I tried to do some research on this, and the closest thing I could find was that it could maybe be either a peripheral dendritic cell or a possible regulatory dendritic cell. And I just broke this down because I know that it's ECR7 positive, which can be on T cells and mature dendritic cells. Unfortunately, I did not have CD11C present. So for my next replicate, I was supposed to use a CD11C. However, with COVID, I was unable to have a few of these reruns. And I just broke this down into my remission phases and what I have for my hypothesized cell populations, which is just seen in the right. And then I just further broke this down to say, okay, I believe that samples 3 and 7 are my CD8 positive T cells, and samples 4, 6, 10, and 13 are most likely some type of CD4 positive T cell. There has been a lot of problems in troubleshooting with this sample, or with these experiments. For my naive brain and spinal cord, I had contemplating needing sample pooling just because I could only get about a few, 10,000 cells, which is not enough if I'm using an equal way to analyze my data, just because then the other samples that I'm getting in the hundreds of thousands for can only be limited by that then. Again, my other samples that I mentioned previously had some debris in it, so they were unable to be used. I've had some staining issues as well with overstaining of cisplatin and CD3, a possible understaining just because of my metacluster 15. I've looked at using digest buffer just because otherwise I will find sometimes my cells get stuck in the myelin layer, and just also using help with data analysis and how to go about it. We have more progress in troubleshooting. So again, with the pooling of naive spinal cords, I'm hoping this will help get rid of my debris. For staining, again, there's more understaining for some of these. I'm going to try and learn how to barcode, which will help keep everything more consistent for my stainings. There were some that did not have any stains, such as a CCR9 or a neuropilin-1, and that may be due to understaining or because there's too much present. There may be and I've looked at my digest buffer time and how to run EAE. I did have a sample that had one bulge, but we had to discard it. So in concluding remarks, further analysis of metaclusters for some markers can be very weak and more replicates are needed. For future experiments, I would like to look at naive samples to better understand my disease phases, and hopefully I can look at just a pertussis toxin only model at the same time increments. And as mass cytometry does not show cell location, I would like to employ immunohistochemistry in order to try and look at this. And altogether, this work will reveal if new cell types exist in MS that could potentially provide a more targeted therapeutics. And thank you so much. I really appreciate everyone coming out and listening to my presentation. I will now look at the questions. So if anybody has any questions, please let me know.
You got one, Kaylyn. Yeah.
Okay. Have you separated the meninges from the spinal cord just to ensure you're looking at the cells in the parenchyma? So what I do for my isolation is I just take the whole spinal cord, we leave the brain and the like everything else, we don't put that in, and then we use the entire spinal cord and then use a digest buffer to just break everything else down. So for CD3E, it's just basically CD3 as well. It's just the way that the markers are labeled for CyTOF. So CD3E is very similar and basically can be used as CD3.
So thank you, Kaylyn. And we'll go forward now with Daniel. Yes, Daniel, we can see your presentation now. So put it on presentation mode.
Hi, sorry, just want to turn on my camera first.
Yeah, okay. So Daniel has just finished his first year in biological sciences at U of C. He has plans to pursue a career in clinical neurosciences. Currently, he's investigating the potential of phase congruency as a novel measure of lesion activity in MRI of MS patients. And he's working currently with Dr. Yoon Zhang. So go ahead, and we're looking forward to hear you.
Thank you, Nita, for the introduction. So, hello, my name is Daniel. Today I'll be presenting about the potential of phase congruency as a novel measure of lesion activity in magnetic resonance imaging of multiple sclerosis patients. So some background information for this project. Multiple sclerosis is an inflammatory demyelinating disorder of the central nervous system, which is characterized by focal lesions in both gray and white matter. So there is currently no cure for multiple sclerosis. There exists a wide array of treatments. The effectiveness of these treatments heavily depends on how early the lesions are detected and it is therefore important to be able to measure multiple sclerosis pathology accurately and early on in the course of the disease to reduce patient disability. Currently, magnetic resonance imaging is the primary imaging modality used to observe multiple sclerosis progression in patients, making it an irreplaceable tool in management of the disease. Fluid attenuated inversion recovery, T1 weighted and T2 weighted imaging sequences are used commonly in clinical practice, and though these sequences allow for accurate detection of white matter, they only detect approximately 30% of gray matter lesions. This poor rate of gray matter lesion detection is particularly detrimental since there is evidence that suggests that 59% of lesions in severe cases of early MS occur in the gray matter, significantly impacting the physical and cognitive functionalities of the patient. It has recently been shown that double inversion recovery and phase-sensitive inversion recovery MRI sequences can increase gray matter lesion detection rate. Double inversion recovery uses 2 inversion pulses to null the signal from both the cerebrospinal fluid and the white matter, which permits detection of twice as many lesions as FLAIR. And phase-sensitive inversion recovery considers the phase of the magnetization of molecules, which increases contrast and permits detection of 2.6 times as many lesions as FLAIR. And though these sequences improve gray matter lesion detection, they still lack the sensitivity desired for clinical use. To further increase sensitivity of these promising sequences, starting post-processing steps can be applied. One issue in particular that can be addressed is the lack of contrast that is often seen in certain areas of brain MRIs, which makes it difficult to detect areas of hyperintensity that signify lesions. One pre-processing method that can significantly improve contrast is phase congruency. Phase congruency is a texture analysis method that is highly sensitive to the edges and corners of a structure, irrespective of image contrast or, in this case, signal intensity. Unlike conventional edge detection modalities, PC employs the K-space or frequency domain data of the MRI image. It detects regions of high contrast based on how in phase with each other the frequencies that represent a certain region of the image are. Phase congruency considers the ratio of frequency magnitudes of the overall magnitude of the point to determine contrast rather than the frequency magnitudes alone, as is conventionally done. As seen in the figure, this allows phase congruency to detect edges and corners equally well in dark and bright regions. So the methods to test the applications of phase congruency in detection of MS lesions, we obtain postmortem MRI scans from 30 MS patients, which are mostly progressive, secondary progressive, and primary progressive patients. Each of the patients was imaged with T1 weighted, T2 weighted FLAIR, PSIR, and DIR imaging sequences. A weighted mean phase map and a sum of phase congruency map was calculated for each sequence. The images were then semi-manually segmented for lesions, twice in image J, once using only the image, and then the second time using both the image and the calculated phase congruency map. The lesions were then classified either as gray or white matter. Area of the lesions was measured in image J, and the lesions were verified using histology data in order to assess accuracy of the method. Statistical analysis was used in order to determine if there's significant differences between lesion number and area sample means. Before use, the phase congruency algorithm parameters must be adjusted for the type of image or size of features that are planned to be evaluated. So in our lab, we adjusted the phase congruency parameters separately for each of the 5 sequences tested. I won't get into the parameters now, but ultimately the parameters were set to allow for detection of the smallest lesions possible while avoiding interference from the noise. So now looking at some of the results that we calculated for the original images and some of the maps that we calculated with phase congruency. So here's the original T1 image. This is a coronal view of the brain, meaning that it cuts through the brain perpendicular to the plane separating the 2 hemispheres. You can see that there's practically no contrast between different tissue types. The only discernible features are the ventricle and some of the cortical sulci present. Now looking at the phase congruency map, we can observe a little more contrast in features. However, the image still appears too busy to identify anything beyond the ventricle and the sulci. The weighted mean phase map, which is another outcome of the phase congruency algorithm, also appears busy. And while it has more contrast than the original image, it doesn't have enough to detect any lesions. Thus, unfortunately, we can conclude that phase congruency cannot elevate contrast, which simply isn't there due to the nature of the imaging sequence. Highlighted in cyan on the last image are the locations of the histology-confirmed gray matter lesions. Looking at the same areas in the original image, we expectedly cannot recognize them as lesions. Looking at the same area on the face congruency map, in retrospect, we can recognize that a lesion, there is a lesion that extends from the ventricle. However, it is not discernible from a similar structure that emerges from the bottom of the ventricle, implying that though face congruency T1 map might be sensitive to lesions, it lacks specificity. And finally, looking at the weighted mean phase map, we can see no contrast between gray matter lesion and the surrounding tissue. Here are the T2 results. Now looking at we can already see some improvements over T1. We can see a region of hyperintensity extending from the ventricle that is most likely a white matter lesion. And we can also more easily observe the sulci and other structures in the brain. Looking at the face congruency map, we get an even clearer look at some of the different tissues present in the slice. The white matter lesion is also more clearly visible on the map. The weighted mean phase map does appear over-selected. Still, the white matter lesion and certain structures are visible, though this map likely doesn't provide a significant benefit over the original image. Now looking at the histology verified gray matter lesion, we can see that though the original image has contrast between white and gray matter, it does not have contrast between healthy gray matter and scar tissue. Same is true for the face congruency and weighted mean phase maps. It is impossible to discern these gray matter lesions from the surrounding tissue. Now looking at the best conventional MRI sequence for MS, FLAIR, we can quickly see some improvements over the previous two sequences. Besides just seeing the white matter lesion and other structures much more clearly, we can see an additional juxtacortical lesion at the top of the hemisphere. Juxtacortical lesions are white matter lesions that are near or oftentimes inside cortical gray matter, which we did not see in the previous two sequences. Contrast between the different structures and white and gray matter is also improved for this imaging sequence. The face congruency map further improves on this contrast, allowing the white matter lesion, juxtacortical lesion, and sulci to really stand out from the surrounding healthy white matter tissue, though it does appear a little busy and over-selected, which might cause problems in identifying the exact boundaries of lesions. The weighted mean phase map for FLAIR appears far too busy, and though the aforementioned features are still discernible, lesions can easily be lost since a lot of white matter appears bright. Now looking at the histology verified gray matter lesions, we can see that FLAIR lacks the contrast necessary to detect these small, exclusively gray matter lesions. The lesion on the tip of the sulcus above the ventricle does appear hyperintense on the face congruency map, but the lesion on the border of the ventricle appears completely black, which shows that the face congruency map is not consistently sensitive to these lesions. The weighted mean phase map is too busy to detect these small lesions as well. Now looking at one of the promising new imaging sequences for face-sensitive inversion recovery, we can right away see that there is significant contrast between white and gray matter, and this allows for much better detection of lesions. The white matter lesion can be clearly distinguished from the surrounding tissue. However, the juxtacortical lesion is not visible. On the face congruency map, clarity and contrast are improved further over the original, so much so that the juxtacortical lesion actually does become visible, which really shows the advantage of the face congruency algorithm. And the weighted mean phase map, though busy, also allows features to be seen. And two of the histology verified lesions are discernible in the original image, and even more so on the face congruency map, and not as well on the weighted mean phase map. The final sequence that we tested, double inversion recovery, also appears much better than the conventional sequences, especially T1 and T2. The sulci are more well-defined than in the other sequences, and both the white and the juxtacortical lesions have contrast with the surrounding tissue have more contrast with the surrounding tissue than FLAIR. Face congruency elevates this contrast further. Healthy tissue appears completely black, which greatly increases the clarity and contrast, allowing for very straightforward detection and segmentation of lesions, which I think is the effect that we're trying to achieve. And the weighted mean phase map is once again too busy, but the white matter lesions are still visible. And looking at the histology verified lesions, we can see that the only one that is clearly visible on the original and the face congruency map, and none of them are visible on the weighted mean phase map. And here are some of the quantitative results that we collected. It must first be noted that these results are not yet histology verified. the lesions considered here are what we assume to be lesions based on experience, but they are not necessarily accurate with the histology. All of the p-values presented here are for paired samples t-test. Please also note that the FLAIR and DIR sequences have 92 slices per patient, while the other sequences only have 5. And as a result, we cannot compare the lesion counts directly between these two groups. And we are currently looking for ways to indirectly compare these. The first table compares the average number of lesions detected using each sequence alone versus using the sequence and the phase congruency map. We can see that there are significant differences in the total average number of lesions detected with and without phase congruency for all sequences except T1. And we see the same results for white matter lesion averages. The averages for gray matter lesions are significantly larger with phase congruency for all sequences except T1 and T2. And these are results that we expected since T1 lacks contrast in both types of matter, and T2 lacks contrast for gray matter. The second table compares the average lesion areas measured using MRI alone and MRI with phase congruency. We can see that the lesion areas segmented were significantly larger with phase congruency for white matter lesions in all sequences but T1, while gray matter lesions areas were unchanged with use of phase congruency for all sequences. And this is likely the result of gray matter lesions being more focal and small, where if there was a difference in volume, it would not be significant. And here are the results comparing the averages between different sequences. So we can see from in the first table that the total average lesion count is significantly larger for double inversion recovery with phase congruency than all other sequences. And we can also see that double inversion recovery alone performs as well as FLAIR with phase congruency and significantly better than FLAIR alone, which supports the evidence discussed in the background. And we see the same results for white and gray matter lesion counts separately. Looking at the next table, comparing the average area measurements taken for FLAIR and DIR, we can see that the only significant difference in areas is between double inversion recovery alone and FLAIR both with and without phase congruency. Double inversion recovery lesions areas are significantly smaller than FLAIR alone, and FLAIR with phase congruency double inversion recovery lesion areas are significantly smaller than FLAIR alone and FLAIR with phase congruency. And this is likely the result of the busy appearance of the phase congruency FLAIR maps discussed earlier. Looking at the third table, comparing the average lesion counts for the remaining 3 sequences, we can see that a clear ranking can be established, PSIR being the best sequence for lesion segmentation out of this group, followed by T2 weighted, and finally T1 weighted being the worst. This conclusion also supports the evidence discussed in the background section. And looking at the table comparing the average white and gray matter lesion areas, we can see that PSIR and T2 have similar white matter lesion areas between the sequences alone and these sequences with phase congruency, while the gray matter lesion areas were about the same size between PSIR and T2, regardless of whether phase congruency was used or not. We can also see that both gray and white matter lesion segmented in T1 images are much smaller than T2 and PSIR. The most important conclusion we can draw from our results is that double inversion recovery paired with phase congruency consistently outperforms the other sequences in both gray and white matter lesion detection. Furthermore, phase congruency improves the performance of every sequence except T1 through improvement in contrast and clarity in lesion areas. And based on the area results, we can theorize that the phase congruency can improve detection of white matter lesion areas. And based on these conclusions, we suggest that phase congruency should be used alongside routine MSMRI scans or in conjunction with double inversion recovery sequence to improve gray matter lesion detection in clinical setting. Further work for this project would consist of evaluating the applications of phase congruency on other imaging sequences and other Tesla field strengths, and further tuning of the phase congruency parameters to further increase clarity and reduce the busy appearance of some of the maps that you've seen today. And the final future project that we can do is we can compare phase congruency to other texture analysis techniques for segmentation of MS lesions. And lastly, I just want to thank the Alberta MS Network for providing me with a studentship that allowed me to do this work. Dr. Yina Zhang for supervision, Olinka Oladosu for creating the face congruency code that I used for this project, and the rest of my lab mates for helping me with various little things. And that is all.
Thank you, Daniel. You can go in the chat box. If you can see, if you can stop sharing.
Oh, okay.
Yeah.
The most surprising finding, probably how much just a post-processing method can actually improve and change how the image looks. So how from what you saw, from what I've shown, you can see that like for some of the sequences, MRI sequences, the change in contrast is very drastic. And any other questions?
Just give them a couple more seconds.
Yep. All right.
Yeah. Wow. Yeah. Okay. One more.
Yeah. How translatable is the processing to clinical work? So I think that face congruency is actually very translatable to clinical work because the method itself is not very resource intensive. So it doesn't take a lot of time to calculate these maps in the clinic, which is important because time is limited in that setting. And also the way face congruencies works. So you just have to tune it for the type of image you have, and then it will work well for all images, for all patient images of that type. So if you have like a T1 image, you just tune the face congruency algorithm for that image, and then running the face congruency algorithm on any image of the T1 type with the settings that you have will produce the same results consistently.
More questions from the audience?
I'd just like to thank Daniel for the great work and commitment in the summer as a first-year student who has just finished the first year in biology. So he's done tremendous work, I believe. Yeah, I thank you for your hard work for switching back and forth things and put everything in order.
Yeah, thank you, Union, for, I was thinking that you might not be able to join us, but.
Yeah, I was jumping here from my other computer to here.
Yeah. So with that, thank you, Daniel, Kaylyn, and Natalie. You all have been wonderful today. And I would like to thank my co-host today, Trisha, and the wonderful audience. You have been a great support throughout this series, and for your wonderful support today for our summer students. And we wish all the summer students all the best for your future endeavors. And we'll see you next Thursday. Bye-bye.
Thank you.
Thank you.
Hi, Trisha. Okay.
Hi, Union.
Good to see you. Take care. Oh.